Good morning, everyone. As you get settled and have a seat, I'll start the introduction of our speaker for today. So, Dr. James Warnock is coming to us from the University of Georgia, where he is the founding chair of their relatively new School of Chemical Materials and Biomedical Engineering. And before he was at UGA, he served as Associate Dean and Professor at Mississippi State University and the Bagley College of Engineering there. And he earned a doctorate and master's in biochemical engineering from the University of Birmingham in the United Kingdom. And perhaps where I first met him, maybe it was time at Georgia Tech, or maybe when you're Mississippi State. I'm not exactly sure when our paths crossed. He also did his postdoc at Georgia Tech under Dr. Yoganathan, for those of you who do that work. So he has conducted research all across the areas of cell and gene biomanufacturing, bioreactor design, tissue engineering, and mechanobiology. And he's going to present today some of the work they're doing at UGA, leading the CMAP program in general and workforce development for cell manufacturing. Thank you, everyone.
Right. Thank you, Josh. So it's great to be here. Thank you for the invitation to come and speak. And it's great to see all the students here this morning. So I'm starting to feel old. Okay, I remember when I was your age, when I was a bright-eyed student, all excited about what was going on, and now, I had dinner with Josh last night, and we were talking about how there were students that we had as undergraduates that are now serving on my advisory board, the faculty at various universities. So I'm starting to feel old. So it's been a real journey, though. Over the past 20 years or so, getting to where I am, and it's been exciting to really see how the field of biomedical engineering has evolved and developed over the course of my career. So with that in mind, I was going to start off by just giving you a timeline of what happened in the field of biotechnology and biomedical engineering in the 20th century. So biotechnology actually has been around since the dawn of time, and probably the oldest biotechnology or biochemical engineering process is fermentation. So when you have that glass of wine or you have a beer, that's a biochemical engineering process that we have. But biotechnology, as it applies to human health care, really took off in the 20th century. In 1928 was when the first antibiotic was discovered. And that was a real breakthrough because certainly during the First World War, one of the main causes of death for soldiers was not necessarily wounds from being shot or explosions. It was the infections that they got as a result of those wounds. And so septicemia was probably one of the leading causes of death. So discovering antibiotics was certainly a major breakthrough. But it wasn't until 1942 that penicillin was able to be mass produced, and that made a huge difference in subsequent conflicts, the Second World War, and other conflicts that we've been involved with since then. I won't go through all of these things that I have on this timeline, but some of the important ones to note was 1953, Watson and Crick discovered the double helix, the structure of DNA. And of course, that then led to the development of the fields of molecular biology and genetics as we started to understand the basis of life. So as a result of that, 20 years later, in 1973, they had the first recombinant DNA experiment where they were actually able to manipulate the genetic material so they could produce a protein of interest. And that led to the production of monoclonal antibodies, which now, of course, are a very important therapeutic. We had, in the early 80s, PCR was developed. How many of you do PCR in the lab on a daily basis? Can you imagine doing research in a lab where you don't have PCR? That's something we take for granted now, but that was developed in 1983. So that's a technology that's less than 40 years old. Then in the early 90s, we had the first gene therapy treatment performed. We had genetically modified foods. We said that we were going to completely map the human genome. And Dolly the sheep was the first mammal to be successfully cloned. And so at this time, at the end of the 20th century, in the 90s, that was when I was a student. Dolly the sheep was a very exciting breakthrough. Gene therapy was a very exciting technology that was going to revolutionize the way in which we treat several different diseases, and everything was looking good. And also during the 20th century, we had huge breakthroughs with semiconductors and microchips and stuff. The internet was born in the 1990s. So there was this whole technology revolution that had gone on around computers and electronics and then biotechnology. And there was a lot of optimism at the start of the 21st century. So the 21st century was going to be deemed the biotech era. And there were all these technologies that we were going to have. Some of these have come to fruition, some of them have not, but all of them have some sort of ethical component to them that we have to think about. So as companies and research labs were discovering new genes, they were patenting them, and there was some controversy about whether a gene can actually be patented and who owns the rights to a gene. I mean, if it's a gene that we all have, who owns that gene? We had transgenic bacteria that were being able to, or were supposedly going to be able to use to reduce waste, help the environment, clean up oil spills, things like that. We had GMOs. Now you go to the grocery store and, you know, several foods are being labeled as genetically modified, and there was questions to whether that should be allowable. Like I said, Dolly the sheep, the first mammal to be cloned. You know, what happened to cloning? You know, we never hear about cloning. That was going to be a huge breakthrough in biotech, and it was a technology that was never able to be successfully commercialized. There was worry about designer babies, and even now we have genetic screening. So if you have a genetic disorder and you're thinking about having a child, you can get the screening to make sure that they're not carrying a certain gene or a certain trait, or what the likelihood is that they will develop that same genetic disorder. But there was concern about how far ethically that should be taken. And then there was also genetic profiling. You know, could insurance companies genetically profile people and then not allow them coverage based on their genetic profile? And we have things like Ancestry.com and 23andMe, where you can now submit a saliva sample and they'll do your genetic profile, and you can find long lost relatives, and then the police are using it to solve cold cases and things like that. So all these technologies were being developed. And I apologize, this is slightly out of focus. You know, Scientific American in 1999 published a special issue on gene therapy. They said that gene therapy by the year 2010 was going to be a $10 billion industry worldwide. As it turned out, by 2010, not a single gene therapy product had been approved by the FDA or the EMA in Europe. So despite all this optimism, some of these things didn't necessarily come to fruition. In May 2000, Time magazine published an issue saying, you know, predicting what would be the hottest new jobs of the 21st century. I believe it was number 2 on the list, was tissue engineer. They said tissue engineering was going to be this whole new profession where we were going to be able to create artificial organs that would help with the organ transplant shortage. We'd have new kidneys, we'd have new heart valves, we'd have new bone replacements, all these things, and this was going to be a huge area. Again, by I think 2005, I can't remember the exact number, but a significant number of tissue engineering startup companies had actually gone out of business. And tissue engineering, again, you know, 2022, we still don't have a single approved tissue engineering product. So despite all of this hype and all of this excitement around these technologies, they didn't necessarily live up to that. But as a result of the work that was being done in these areas, it did lead to other things that have now been successful, and the technology has led to some really significant advances. And one of the main outcomes of this has been the growth of the field of cell therapy. So if you do a Google search, you'll find that there are a number of different definitions for cell therapy. This is the one that I like the most, but it's not definitive, but I think it's the most, to me, it's the most descriptive. But it says, cell therapy is the prevention or treatment of a human disease by the administration of cells that have been selected, multiplied, and pharmacologically treated or altered outside of the body or ex vivo. So in cell therapy, what we're doing is the cell is the drug. In other types of biologics, we have monoclonal antibodies, we have recombinant proteins, we have vaccines that are produced from viral vectors, we have chemically synthetic drugs, we have all types of different drugs. With each one of those drug classes, the drug can be thought of as a non-viable static entity. The difference with cell therapy is that the cell is a living entity, and as you know, the cells, because they're living, respond to any environmental cues that they have. And that means that we face a number of challenges when we're trying to use cells as a therapeutic, because you want the therapeutic to be consistent. You want to have quality assurance that it's not going to change, it's not going to be altered. You want to have some control over that process so that there is reproducibility, so that every patient receives an administration of the drug. You can be confident that they're getting the same dosage, that they're going to have the same therapeutic reaction to it. So the two primary cell therapies that are being investigated today, the first are stem cells. And so stem cells were the product of tissue engineering. In the early days of tissue engineering, hopefully you're all familiar with tissue engineering, but if you're not, tissue engineering basically is where you have a scaffold material that mimics the extracellular matrix. It can be made of a biologically derived material such as collagen or a hydrogel, or it can be a synthetic material like PLA or PGLA. That scaffold supports growth of cells, and then the premise is that the cells will differentiate, or if they're already differentiated, they will start to grow and produce their own extracellular matrix, and they will develop into a living tissue. And so a very simple, basic tissue engineered construct would be skin. It doesn't have much complexity to it as a tissue, compared with something like a liver or a kidney that's performing a lot of metabolic functions within the body. So tissue engineering originally was based on using differentiated cells. So if you wanted to grow bone, then you would use osteoblasts and osteoclasts. If you wanted to grow a blood vessel, you'd use smooth muscle cells and endothelial cells. But that was presented a lot of technological challenges, and so as stem cells, certainly adult stem cells as opposed to embryonic stem cells, were starting to be investigated. They became of interest because it was felt like those stem cells could then be used as in tissue engineering to regenerate the tissues and differentiate into the particular cell type that you wanted. The challenge was, how do you get the stem cell to differentiate? And you can use mechanical cues, and you can use biochemical cues to do that. One of the interesting outcomes of that was that it was discovered that stem cells actually will secrete various different biomolecules, which is referred to as the secretome. And the secretome, along with cell-to-cell contact, can regulate the immune system. And so now, whereas, stem cells have been investigated in regenerative medicine for several years, now stem cells are being used for immunomodulation, where the stem cell is administered to the patient, and it's called the hit and run effect, where the stem cells will be administered, they secrete these biomolecules, it stimulates the immune system can then attack whatever the patient is suffering from, and then the stem cells will disappear. And so because they have this immunoregulatory effects, the International Society for Cell and Gene Therapy officially changed the name of mesenchymal stem cells to mesenchymal stromal cells a few years ago. So when you see the acronym MSC, that now refers to mesenchymal stromal cells as the official title of that. So MSCs have been or are being investigated a lot as a cell therapy, certainly as a way to treat cancer, solid tumors. There's still a lot that is unknown about MSCs, and there still aren't any MSCs that have been approved by the FDA as a cell therapy. But one of the things that we are sure of is that when you have an MSC, in order for it to have some sort of therapeutic benefit, you need to produce about 10 to the 9 cells for each administration. So 10 to the 9, we're talking about 100 million cells that we're trying to grow. In doing that, we need to make sure that the MSCs are producing the biomolecules of interest, that they're not differentiating, that they're maintaining their viability, and that their therapeutic effect is reproducible and reliable. The other key cell therapy that we have are CAR T cells. Let me see if I can play this. All right, let's start this from the beginning.
If it's working properly, the immune system wants to kill cancer cells. That's its job. Create an army of T cells to patrol our body for signs of infection and other diseases. The T cells inspect suspicious cells and, when they find an abnormal one, immediately lead an attack on it, while limiting the damage to healthy cells. The way they do that is pretty elegant. T cells have protein receptors on their surface, and these claw-like structures latch onto antigens, protein fragments found on the surface of all cells. When a receptor latches onto an abnormal antigen, like one found only on a diseased cell, the T cell turns on and releases toxic chemicals that damage the abnormal cell and recruit other immune helpers to the area. Sometimes cancer cells develop ways to fool the immune system, either by disguising themselves as healthy cells or by sprouting so many antigens on their surface that the T cells are stymied and can't mount an effective attack, or by finding ways to turn off the immune response against them. One innovative way to see through the deception is CAR T cell therapy. Thousands of a patient's own T cells are collected in a process similar to a blood donation, using a modified and inactive virus that can't cause any disease but can introduce genetic information into a cell. The T cells are reprogrammed so they now produce special receptors called chimeric antigen receptors, or CARs, on their surface. These newly engineered cells, called CAR T cells, are grown in a lab, and millions of them are infused back into the patient. The new receptors enable them to latch onto a specific antigen on the patient's tumor cells and destroy them.
Okay, so CAR T cells are the direct result of gene therapy. So gene therapy When I did my PhD, I was working with a gene therapy company in the UK, and they had retroviral vectors, they had lentiviral vectors. They were developing so that they would be safe and effective, but then those viruses would be administered to a patient, and then they would kill the tumor within the patient. That technology had a lot of unintended clinical consequences, and that is why a gene therapy, a pure gene therapy like that, was never approved. I'll tell you about. There was one gene therapy that was approved in Europe, but it was for a very rare disorder, and so it never had a huge market demand and was extremely expensive, and so it was never approved by the FDA. Yes.
What were some of the consequences that you mentioned about the gene therapy that was used?
So some of the consequences are an immune storm, where you have this huge immune reaction. Another consequence is that depending on the type of virus that you're using, it has a transient expression. So if you're using an adenovirus, you administer that to the patient, and then the body clears it very quickly, so it doesn't have a chance to have a therapeutic effect. So ultimately, there are also concerns about safety. So if you have a virus like a retrovirus, and that gets administered to a patient and it becomes intracellular, then it could combine with the patient's own DNA, and then that could cause mutations, and that obviously isn't what you don't want to happen. You want the patient's cell to be able to allow the virus to reproduce its therapeutic genes and produce the proteins that cure whatever disease it is. So there are a number of safety issues and efficacy issues as well. Okay. But what gene therapy allowed us to do was to develop lentiviral vectors that produce the CAR-T antigen. And so now, rather than injecting the viral vector into a patient, as you know, the drug itself, with CAR-Ts, we extract cells from the patient, then we transfect them with the virus, and then once that expression is stable, they can be expanded and then given back to the patient. The first CAR-T cell therapy was approved in 2017, and since then there have been another 3 CAR-T cell therapies that have been approved by the FDA and have been approved in Europe and Asia as well. And so this is a field that's expanding very rapidly. At the moment, CAR-T cell therapy is used for leukemias and non-solid tumors, but there's a lot of work going on about how we can develop this so that it can also be used to treat solid tumors. Okay, so I mentioned with the MSCs that we need to produce 10 to the 9 cells per patient. With the CAR-T cells you saw from the video, once they've been transfected with the viral vector, they then have to be expanded ex vivo to achieve a high enough number that they can be re-administered back to the patient and have a therapeutic effect. So the manufacturing of these cells is a big part of the process. And certainly in clinical trials where you have a small patient population, the manufacturing can be done at a lab scale. But, as we all probably learned from COVID, when you have a large population, manufacturing at a large scale can become a real bottleneck. And, when the vaccines for COVID were first developed, Pfizer, Moderna, and AstraZeneca, and Johnson & Johnson, they had these great vaccine products and they were able to show their effectiveness in clinical trials. The challenge that came after that was how do they produce enough vaccine to have sufficient number of doses for the entire country or the entire world, global population. And so manufacturing then became the key issue. And it's the same with cell therapies. How do we scale up the manufacturing so that we can produce enough cells? But also, as I said before, the cell is a living entity. So how do we produce enough cells and be confident that what we're producing meets certain quality control standards? And so the big question here is, how do we achieve reproducible, low-cost manufacturing of therapeutic cells at an industrial scale while ensuring consistent and highest predictive quality? And so in order to address this question, in 2016, the National Cell Manufacturing Consortium was formed. And this was a public-private partnership, and it had a number of representatives from industry. 16 different universities were part of this consortium, as well as the FDA, NIST, DoD, NIH, and NSF. And this consortium went about developing a roadmap. And the first roadmap that they published was in 2016. So that was prior to the FDA approval of the first CAR T-cell therapy. But their roadmap basically was mapping out the barriers to being able to have a reproducible, sustainable manufacturing process for these cells. That roadmap then became the basis for the proposal that went to NSF for the Engineering Research Center in Cell Manufacturing Technologies. That research center was then funded in 2017, and the consortium then revised their roadmap in 2017, now that the FDA had approved the first CAR T-cell therapy. And then again in 2019, this group, this consortium, came together again and published or developed a third roadmap that had been updated based on the advancements that had been made in cell therapies, over the prior 2-3 years. So the roadmap is available. If you can go to this website here, cellmanufacturingusa.org. That is actually the website for the CMAT Engineering Research Center. But the roadmap is on there, and it breaks down the challenges in various technical areas, how we get reproducible, scalable manufacturing, the challenges with identifying critical quality attributes for ourselves, so we know what to measure in order to tell us that these cells are reproducible and they are doing what we want them to do. The challenges in developing the technologies in order to be measuring the CQAs. But also one of the important things that's in here is how we have a workforce that can work in the manufacturing area in order to produce these things. Because we can have the best technology in the world, we can have the best research in the world, we can have the best manufacturing facilities in the world, but if we don't have a trained workforce that can actually perform the tasks that need to be done, then things will just sit idle. Just a side note, I've done a lot of work with some universities in China pre-pandemic, and China is a fascinating country to visit, and the government is investing a lot of money in different technologies. They obviously want to be a world player and be competitive on an international scale. And so they've invested a lot of money in these great facilities and these great research centers. And, I've visited some of these kind of campuses, which is like a city within a city that's just space for startup companies and research facilities. And you walk around and they have the latest technology, the latest equipment. But the thing that you always notice is there are no people. And the challenge that they have in China is not developing the technology, it's developing the workforce. And, it sounds a little bit counterintuitive because China has such a large population. But so they have the potential, but it's training those individuals. And that's the same challenge that we face with cell manufacturing. We can have the best facilities, but we need to have a trained workforce to do the work as well. So, all right. So within our roadmap, I was part of the group that worked on workforce challenges, and there were 5 areas of 5 core challenges that we identified in workforce development. And I'm not going to focus on all 5 of these. They're all important. The 2 I'm not going to worry about today are the communication about cell manufacturing careers. We need to be building a pipeline. That pipeline very often starts in middle school and then high school. You know, we need to be getting these, you know, young students interested in this field, so that's something they aspire to. And then, you know, we want to have a diverse and inclusive workforce because, you know, that brings different perspectives, which creates a much richer environment. I could probably talk for hours on the importance of diversity and inclusion within the workforce that we're developing. But today, I'm just going to focus on these 3 areas, and then they all kind of work together. So at the moment, we don't have any official educational programs related to cell manufacturing. You know, we have degrees in biomedical engineering, we have degrees in chemical engineering, we have degrees in industrial engineering, we have degrees in molecular biology and biochemistry, but we don't have anything that's specific to cell manufacturing. And so that was seen as a core challenge. How do we create educational programs specific to this field where we can train people with specific skills? There's a lack of multidisciplinary skills across the manufacturing workforce. This is something that our industry partners highlighted. You know, there are people that are very well trained in doing tissue culture and culturing cells, but it's all done at the bench scale. They do T-flasks. And when you're trying to produce an industry scale number of cells, if all you can do is a T75 flask, that's a lot of flasks that you're going to have to use. On the other hand, you have people that are familiar with using 10,000 liter bioreactors, but they have no idea how to culture tissue culture or mammalian cells in those environments. And then there's also, you know, how you introduce omics and molecular techniques, mathematical modeling and machine learning and data science. You know, there's a lot of multidisciplinary skills that are required in this field. And then also there needed to be, you know, one of the things that we identified as a challenge was, you know, industry wanted one thing and academia was producing another thing, and there was no crosstalk between the 2 of them. So we wanted to make sure that what we were doing in academia was really meeting the needs of industry, because ultimately they're the ones that are going to be hiring and employing the workforce. So, you know, the skills that we said our workforce needed fall into the technical areas and the professional areas. So the technical areas, scalable cell processing, process integration, omics and modeling, and sensors and controls. You know, those are the skills we need in order to develop large-scale manufacturing and also measure the quality assurance of our product. But then also we said it's not just enough to have skills in these technical areas. Our workforce also needs to have skills in these professional areas as well. So, you know, I mentioned at the very beginning of my talk the ethical issues associated with a lot of the 21st century biotechnology areas. So we certainly want our workforce to be aware of the ethical aspects and policies surrounding what they're doing. Yes.
Can you clarify who's asking for the workers to be knowledgeable in ethics and policy? Is that academics, or is that industry, or is that taking a step back and ideally they would have it?
Yeah, so yeah, these areas were developed as part of our strategic plan. And so it was really a combination of feedback from industry, from our industry partners, and also you know, academics saying, you know, we think this is what should be included. But yeah, and you know, cultural sensitivity as well it was felt like if you're going to work in a diverse and inclusive workplace, if you're going to work in a global workplace, you had to have some awareness of different cultures so that you could work in an interactive way, collaborative way there. And also, you know, these last of the industry perspective and the regulatory knowledge, you know, we certainly wanted the workforce, you know, we're creating a workforce, but most of the workforce have never worked in the industry. You know, maybe they did an internship as an undergraduate, or they did a co-op, but they don't have extensive experience in industry. So we wanted to make sure they at least had some perspective of what industry was like before entering the workforce, and what some of the constraints were in terms of regulations surrounding these things. You know, what the FDA requires, what NIST may require in terms of standards for manufacturing and things like that. So these areas are fairly broad, and so we wanted to take these areas and make sure that we had defined certain competencies that our trainees would have. So we got our input from our constituents. You know, we first of all had identified these areas. We got our input from our constituents, which were a combination of academia and industry. Those results were reviewed by our education and workforce development team. And based on that review, we recommended certain competencies that we thought trainees should have based on that input. That was presented to our executive committee. In an ideal world, the executive committee would have said, this is great, no further changes are needed, we would have had our set of competencies. But this is all an iterative process. And so in reality, the executive committee said, you know, there are some things here, this is good, this is a good start, but there are some things that need to be changed. So we took their additional comments, we recommended some changes to those competencies, and we went through this process. And like I said, this is an iterative process. We could have gone through this multiple times. In reality, we only had to make one set of revisions before the executive committee agreed to what we developed. And then we have our list of training competencies.
Yeah, question?
Yeah.
Did you ever go all the way back up to the constituencies and ask them if, what their, you know, ask them to comment on what your final recommendations were? We, at this stage, no, we haven't. But that's because we're still, I mean, this process is something we've been working on for the past, maybe the past year, maybe a little bit longer than that. It would be beneficial, certainly within the next 3, 4, maybe 5 years, to go back to the constituents and say, are these competencies still relevant to what we're doing? Because the technology is going to change between now and 2025. So we want to make sure that, you know, if anything does change, that we can be agile and adapt to that. But going back and, you know, having that constant review cycle would always be very important. So when we talk about competencies, this is just, you know, quickly how we define it. We want the competence to be something that's specific. So those technical areas and professional areas are very broad, but we want something specific and measurable so that we can actually assess how well our trainees are doing. And it needs to identify the performance or the skill that we want those trainees to have. And we also said in some way it should be specific to what they're doing within our cell manufacturing technology ELC. I'll just skip over this. So, you know, when you're developing a competency, you want it to have 2 essential parts. You want to have an action verb that really directs the performance of our trainees, so we know what they should be doing and how they should be performing. So they should be able to evaluate or analyze or design or something like that. That action verb should also be at an appropriate cognitive level for the trainees. In this case, we're talking about graduate students or graduate trainees. And so it should be a higher level cognitive skill. So it should be, ideally, it should be beyond just an understanding or a knowledge of something. And then the second aspect, a competency should be the subject of that action verb. So if they're analyzing something, what is it that they're analyzing? If they're designing something, what is it that they're designing? And so on. So we conducted a faculty workshop with approximately 20 faculty involved and broke them down into small working groups. They did some brainstorming, they did some discussions, and each of those working groups then came up with a set of competencies. We then did a focus group with our industry partners, and that was actually facilitated by our Workforce Development Advisory Board. And then we did an affinity process, which is basically taking all this input that you have from these different working groups, from faculty, from focus groups, from industry, and saying, okay, what are the common areas? What are the things, the key things that they've each said, and can we kind of combine those without losing any of the specificity, but can we combine those to break down into a manageable number of competencies? And then, like I said, we presented that to our executive committee, and based on their feedback, we revised that. So these are the technical learning outcomes that we developed. There are 7 of them in total. The first 2 really related to statistical and data analysis. So industry certainly thought it was very important. Students were able to design experiments, and that when they did the experiments, they knew why they were doing it, and they understood what the data actually meant and could apply it in a real way. And industry has a very different perspective on research than academia does. When industry does research, it's because they want to know if they have a product that can be commercialized. They want to know what the outcome is. They want to know how the data can be used. In academia, the purpose of doing research very often is to write a publication. In industry, they don't care how many publications you have or what impact factor they have. They just want to know, how does this help us solve our problem? So very different perspectives to the research. So industry and academia both thought that students should have some statistical training. Industry said we really want students to be able to design experiments. Academia said, you know, students really need to be able to do univariate and multivariate statistical analysis of the experimental data to really draw meaning out of it. The second 2, or the second 3 probably, were really related to scalable manufacturing. You know, what is the difference? What are the constraints when you're trying to do something at a large scale or an industrial scale versus in the on the bench? And so, you know, what's the difference between doing a single cell analysis versus a bulk or a population analysis? What's the difference between bench scale and industrial manufacturing? How can you, what assays should you be doing to monitor for critical quality attributes, CQAs, during the manufacturing process? And then, you know, how do you distinguish between cell type specific constraints using different manufacturing platforms? And then the last 2 were more kind of industry related. You know, do you understand project management? Do you understand supply chain models? And how do these issues really relate to regulatory affairs and good manufacturing processes? Then we had our professional learning outcomes, you know, and these really tie in very closely with those areas. They're a bit more specific than the areas that we had, you know, understanding current and past legal, political, ethical, and social issues, demonstrating cultural sensitivity, being knowledgeable of both international and domestic regulatory policies and ethics, but also one thing that was very important, this should probably be first on the list, but communicating scientific and technical information to various audiences. You know, I think, again, COVID has served as a good example of this. You know, a lot of the messaging that has come from the CDC, the FDA, from the White House during the COVID crisis has been very mixed and very confusing to the general population. And so with cell therapies, we want to make sure that our trainees are able to communicate what they're doing in a very effective way. Now, of course, we have to train our students to be able to do these things. This is actually adapted from a fairly, I wouldn't say old, but an engineering education paper that was published by Pat Terenzini about 20 years ago. It just talks about how students develop their competencies or their learning outcomes. And so, you know, in graduate school, everyone comes in with a different experience from an undergrad based on whether they did undergraduate research, whether they were at a public school, a private school, whether it was a large school, whether it was an R1 university versus a regional liberal arts college. You know, you all came in with a different experience, and that kind of forms a foundation for your learning as you enter grad school. But there are three things that really influence your learning in grad school. There's the curriculum design, what courses you take, what courses you're required to take, the classroom experience, you know, how those courses are taught, a seminar versus a lab class, whether your instructor is using active learning or it's didactic lecture, and then the co-curricular activities, which, you know, a lot of times the research that you're doing in the lab, but can be other things as well, like internships. All those three things contribute to the learning that you have. So we thought about what do we need in order to enable our students to develop their competencies. And so we developed a number of different educational approaches, and we've divided these down into classroom learning and experiential learning. So we've developed two courses specific to CMAT. One, Cell Manufacturing and Society, which really addresses those professional skills, and then one in Cell Engineering and Manufacturing, which addresses the technical skills. And we've also developed a number of course modules that can then be, they're kind of like a plug and play that can be inserted into other courses that other faculty may be teaching. And then we have our experiential learning aspect, the mentored research. Obviously, that's what you're doing in the lab. But then we also have opportunities for students to do industry internships. And we have global exchanges with universities in Canada, in Ireland, and in Japan. So there are opportunities for students to do that. You know, that obviously helps with their technical competencies, but also if you're doing research in a global university, it helps with that cultural sensitivity and really understanding some of those diversity issues. So just to summarize, you know, the way in which we develop these competencies to allow us to understand how we can educate our future workforce in cell manufacturing. We started off by seeking our constituent input. We spoke to faculty, we spoke to our industry partners, you know, and unofficially we've also communicated with government agencies as well, with NIST and FDA specifically. You know, we drafted those competencies, and then we revised them based on the additional feedback that we got from our executive committee. Then we mapped those competencies to learning opportunities, the classroom experiences, and the experiential learning. The next stage will be to assess and evaluate the attainment of those competencies. So we can say this is what we want our trainees to achieve. We can say this is how we are going to give them an opportunity to achieve those. But unless we do the assessment, we don't know whether they're actually attaining those things or not. And so we're now at the stage where we're developing our assessment techniques to really understand how the trainees are developing these competencies. Once we get that feedback and we evaluate the feedback on how they're attaining those, we can then utilize that to improve our educational programs. We can look at ways in which we may want to enhance our courses. We may look at other experiences that students might need in order to have more experiential learning. Obviously, the global exchanges is something we haven't been able to do for the past 2 years because of travel restrictions. But we can look at those and see how these are all playing in and all contributing to the development of the competencies. So that just is to summarize everything that I've talked about. And that's it. So thank you for your attention.
Very nice. Thank you for this very educational training. Training. So it isn't clear to me if this is a undergrad curriculum.
So this is for our graduate trainees.
Graduate.
Yeah, and this is something that we have. I mean, we focused on our graduate trainees primarily because they make up the largest bulk of the trainees in our ERC. We do also have undergraduate students that get involved with research, and we have outreach programs with high schools as well, and also high school teachers. But in order to be focused on what we're doing, we've just primarily focused on our graduate trainees. And the next step of this, I said one of the core challenges, we don't have any credential programs. So the next stage of this is we can take those competencies and we can say, okay, well, we have these 2 courses that we've developed specifically, but if we were to create maybe a certificate program, what other courses would we need to include in that, or what other experiences would we need to be including in that to kind of certify it, to formalize it, so that we can be sure that our graduates are getting these things, and then the goal always is when you have an ERC, that when the funding ends after 10 years or 5 years, hopefully 10 years, the goal is that you want everything to be sustainable. And the research is very, I wouldn't say it's easy because it's never easy to get research funding, but with the research, hopefully the funding has led to more grant proposals and more funding so the research can continue. The educational aspect is a lot more difficult to be sustainable. So if you could develop an actual certificate program or a degree program or something, that helps with that sustainability.
And that accreditation would be a new more accreditation, or would be working on some existing accreditation platform?
So we're not talking about accreditation, we're talking about a certificate. So I'm not sure how FIU does minors and certificates and things like that. In the university system of Georgia, because we partner with Georgia Tech on this, you can have a certificate program that is comprised of, it's typically 15 to 18 credit hours. It's very similar to a minor. It's a lot more challenging to have a minor in a graduate program, but a certificate program is a lot easier. And the other advantage of a certificate program is it can be offered to non-degree seeking students. So if industry has people that they want to be retrained, they can come to the university or enroll at the university. They don't have to be enrolled in a degree program. They can take 5 or 6 courses, and then at the end of that, be awarded a certificate. Now, being a degree.
I have maybe a couple. So you talk about doing this affinity analysis between academia and industry. I'm assuming NSF is probably part of that conversation as well to help determine the competencies that are there. So looking for things that kind of overlap, are any of these 3 bodies ever at odds with each other that you have to balance that? So that's one question. The second question is, when you get into the details, how granular do you go with some of this in terms of, the current needs based upon the current technology?
Yeah. So all right. So the affinity process, you want the competencies to be specific, or to point to specific things, and so, but you also want it to be broadly applicable to all the different areas that you work in. So we have 3 different thrusts within the center, looking at manufacturing, identifying quality control attributes, and developing technologies to do non-destructive measurement of the CQAs. And then within that, we have we have 3 test beds. We have the MSCs, we have the CAR T cells, and we have cardiomyocytes. There's a third cell therapy that I can really talk about today. So it's very broad and very, very deep. So the competencies have to be applicable across all of those different things. So when we're doing the affinity process, you very often, or what we found was there were things that were very specific that were different between, industry may have said one thing, academia may have said one thing specific to a thrust area, and then they may have said something very specific to another thrust area. And so what you're doing is you're looking for that commonality. How do you combine what they said in this thrust area, in industry and academia? What's the common thread between all of that? And that's where the challenge really comes in. And I mean, some of the working groups that we had very specific input that they said. And once you kind of dig through that and figure out, okay, what do they really mean by this? Then you see that thread is the same as a thread over here, and then you can piece it all together. And when you do that, we actually found that there was a lot of commonality between what academia was saying, what industry was saying. And if there is an outlier, then I mean there were a couple of outliers, and we said this is not applicable across all of our center, and so those were left out. We actually, I think my list had 7 competencies in technical areas. That came down from a list of about 15 or 20 competencies originally. And you want to make sure that you don't have so many competencies that it becomes unmanageable. Because we want to be able to assess these things. More things you have to assess, and then that may become distracting. So that's how we. What was the second part of the question?
I think you kind of get them both.
Okay.
Yeah.
All right.
What's an example of one of those competencies?
So the competencies I showed, I can. So these are our technical competencies, or technical learning outcome. We use the phrase competency and learning outcome interchangeably, or we may say student outcome. At the undergraduate level, if you're familiar with ABET terminology, they refer to these things as student outcomes. So these are our competencies. So, like I said, the first 2 are related to statistical analyses. The second 3 are related to the scale-up issues, and then the last 2 are more the industry perspective things. So they're there. Yes.
I just had a couple questions. So the process, you're just in the development phase, right? Like planning on how to train, because I understand that you need like a highly specialized people to do the job.
Yeah.
And I also understand that you want to like mass manufacture it. But my question is, if we're talking about our own cells, right? How can you go from mass manufacturing T cells for the population, for example, right? Or Humira cells, if every individual person is different in the sense of genetics, right? How can you, isn't it just easier to have like this model in which you take the mesenchymal cells from the person and then, you store it, for example, right? And then when the person needs it, because that's what I'm having an issue with, is like the demand of the people. Because I understand that you want to get all these people who have this like supply of workers translates to the demand that's expected in, let's say, the next 5-10 years.
So we want to have a trained workforce that can do the manufacturing. How you manufacture the cells is going to be dependent on whether you have an allogeneic cell or an autologous cell. And so with CAR T cell therapies, that's an autologous cell. You get the cells from the patient, you genetically modify them, scale them up. And so that manufacturing process isn't going to translate to an industrial scale in terms of it being a 10,000, 20,000 liter bioreactor where we're growing the cells. Those are the processes that they have for producing vaccines and monoclonal antibodies and things like that. It's not going to translate to that, but the scale up is going to be more of a scale out issue. So instead of having one large bioreactor, you would have multiple smaller bioreactors. And so it may not be done in a stir tank reactor, it may be done in, say, a hollow fiber reactor or a packed bed reactor, or something that can produce very high concentration of cells, but in a very small volume, because you're only doing it on a per patient basis. But even if you're doing it it would be a small scale reactor on a large scale number of units. So you want to have that. You want people that can do that and do it in a reproducible fashion. With the mesenchymal stromal cells, the gold standard there is really having allogeneic cells. So you don't have to harvest cells from each individual patient. It can be more of an off-the-shelf type of product that you have. And that the challenge is going to be more of a scale up rather than a scale out issue, where we want to be producing these probably not in a 10,000 liter scale, but maybe 100 liter scale. But whenever you're producing these cells at a large volume, whether it be scale up or scale out, you still have to make sure that it's reproducible and the process isn't influencing the cell characteristics in a detrimental way.
Would it be convenient to, let's say, partner up with a company that produces anti-rejection drugs, since we're talking about inserting a foreign body?
Yeah, we have a number of industrial partners that are part of the center. I think we have maybe 35 industry partners, and some of those are contract cell manufacturing companies. Lonza is one of our industry partners. Lonza has a huge manufacturing facility up in New England. They're a global cell manufacturing company, so they have that experience with antibodies and vaccines.
Thank you.
What were the three cell types that you have on that?
The three cell types?
Yes.
We have mesenchymal stromal cells, CAR T cells, and cardiomyocytes.
All right, let's thank everyone up again.